Okay, well, it's February already. Welcome back to Forest Talk, where we talk about the plants and the animals and the connections we have to our ʻŌhiʻa forests. Many of you have requested an update on our endemic ʻŌhiʻa here on Kaua'i, especially in light of the threat of rapid ʻŌhiʻa death. So here it is. Today is the day. And we've been looking forward to sharing this news with you because we have some exciting news that we all are excited about, and how we can move forward in protecting ʻŌhiʻa from the threat known as rapid ʻŌhiʻa death. Or you may hear it referred to as ROD. Oftentimes we shorten rapid ʻŌhiʻa death to ROD. Today's talk is Back Off Beetles, how repelling Ambrosia beetles helps protect ʻŌhiʻa forests. And our guest speaker is Dr. Kylle Roy. Just admitting a couple people. There we go. But before I turn things over to Kyle, I do have a few announcements that I'd like to share. First of all, my name is Kim Rogers, and I am an outreach specialist with Kauai Invasive Species Committee. My focus is on ʻŌhiʻa and protecting our native forest ecosystems from primarily rapid ʻŌhiʻa death, but other forest threats as well. Remember, this is my little plug. Healthy ʻŌhiʻa forests help prevent erosion and ensure we have healthy ocean reefs in which to swim, surf, snorkel, fish. A healthy ʻŌhiʻa forest helps ensure we have fresh water to drink, helps protect our watersheds, and to help stop the potential spread of rod and other threats. We ask that before and after you go hiking or birding or whatever it is you might be doing in the forest, that you practice good biosanitation. And by that, we mean brush mud off your footwear, your gear, whatever it is that might get muddy that you take with you into the forest. That's the first step. And the second step would be to spray that with isopropyl rubbing alcohol. It's the rubbing alcohol that will actually kill the pathogen. So during our time together, if you would practice good Zoom etiquette, that is, mute yourself. If you would, if you have questions, please drop them in the Q&A. That might be easier for us to find than if the chat gets chatty. And we will take Q&A at the end of our presentations today. We are recording, so if you have to leave early or if you have a colleague that you want to share this with who's not able to attend today, I will be sending the recording out later this week. And our next Forest Talk, mark your calendars, will be Wednesday, March 25th at 10 a.m. And my KISC colleague, Halen Chalk, will share, the title of her talk is From the Mountains to the Sea, the Importance of Limu in Connection to Watershed Health. So again, that's going to be March 25th at 10 a.m. And I will also drop in the chat at some point a link to all of our previous Forest Talk sessions, of which there's been, I think, 9. Um, we've talked spiders, we've talked snails, we've talked seabirds, and forest birds. And a whole bunch of different plants. Like I said, I'll drop that link in the chat in a minute. Um, and before I turn things over to Kyle, I do want to take a moment and share a little bit of some backstory and a little bit of an update on ʻŌhiʻa health, rapid ʻŌhiʻa death here on Kaua'i. So I am going to start with a little presentation. So give me a second while I share screen. And Kyle, are you seeing my presentation? Can you give me a thumbs up or not? Yes, we are. Okay, great. So the first thing I want to do is just to give you the succinct backstory, why ʻŌhiʻa is important, and what the threat of rapid ʻŌhiʻa death is. I do have a video that I'm going to play that will keep me on task and keep me on time, and it's going to run about 5 minutes. So once I get it started, if Kyle, you would give me a thumbs up that you're hearing everything okay.
After a fresh lava flow, ʻŌhiʻa seeds, smaller than a human eyelash, find their way into cracks in hardened lava, becoming one of the first plants to take root, thus starting a new ecosystem. A biodiverse forest of tall canopy trees, understory plants, ground cover, ferns, and mosses. This multi-storied forest pulls moisture from fog and catches rain as it falls, allowing it to seep into underground aquifers, the source of our drinking water. Forest vegetation also helps clean the air we breathe, and its leaf litter and complex root system hold soil in place, helping prevent erosion and runoff, ensuring a healthy ocean reef for fishing, surfing, and swimming. A native ʻŌhiʻa forest is more than just a collection of plants. It's home to birds found only in Hawaii, and native beetles, butterflies, damselflies, and tree snails, many endangered. Some of these species have rich mo'olelo, stories, connecting them to the people and places of Hawaii. Many co-evolved with the forest, creating symbiotic relationships with plants. Kahuli, Hawaii's native tree snails, eat mold growing on ʻŌhiʻa leaves, cleaning them and providing greater surface area for photosynthesis, a process that generates oxygen for us to breathe. It's no exaggeration to say almost everything found in our native forest relies on ʻŌhiʻa for life. Throughout generations, ʻŌhiʻa has touched all parts of life in Hawaii. Its strong wood is used for posts, flooring, and once hale, ba'a, ki'i, and kapa beaters. Its flower, lehua, used for medicinal purposes and woven into lei. ʻŌhiʻa are celebrated in hula, oli, and mele. ʻŌhiʻa is our most abundant native tree, and without ʻŌhiʻa, most of our native forests would be unrecognizable. Embodied in its very name, ʻŌhiʻa is the essence of gathering and collecting, as in moisture from the air, as reflected in this saying, Kaua noe lehua o waiʻaleʻale, the misty lehua rain of waiʻaleʻale. A fungal disease known as rapid ʻŌhiʻa death is killing Hawaii's most abundant native tree. ʻŌhiʻa are infected through open wounds, possibly due to peeling bark, torn roots, or broken branches. Microscopic fungal pathogens enter the wound, and the fungus grows inside the tree, preventing the flow of water. The first external symptom is the sudden browning of the leaf canopy. There is no known cure, and after a few months, the tree dies. Then Ambrosia beetles, nature's decomposers, arrive to break down the tree. They burrow into the sapwood where the fungus grows, releasing fungal spores into the environment through their excrement, which can blow to another tree or fall into the mud and be transported on shoes or vehicle tires from one part of the island to another. If these spores reach another wounded ʻŌhiʻa, the whole process begins anew. By practicing a few protocols, we can help stop the spread of rapid ʻŌhiʻa death. Before and after entering the forest, brush mud off your shoes and spray with isopropyl rubbing alcohol. Wash muddy vehicles. Avoid wounding ʻŌhiʻa. Don't move ʻŌhiʻa, dead or alive, around the island. Support the protection of remote and pristine forests. And most importantly, love ʻŌhiʻa.
So, for any of you who recognize the voice in that video, pop the name in the chat. The first person, I'll send you a special sticker if you don't already have one. That probably voice is only recognizable to people on Kaua'i. Not necessarily true. If you're a big KKCR listener, you might recognize the voice too. Um, so let me just take a breath here. Let's talk a little bit about rapid ʻŌhiʻa death rod here on Kaua'i. But before we do that, we have to acknowledge the situation on Hawaii Island. Um, and this is a pretty graphic before and after, if you will. So just preparing you for this. This is a photo of a healthy native ʻŌhiʻa forest in the Puna district on Hawaii Island in 2005. And a mere 10 years later, that same forested area looked like this. So it's pretty devastating. And we estimate over 1 million ʻŌhiʻa have died of rapid ʻŌhiʻa death on Hawaii Island. And unfortunately, that number continues to grow. On Kaua'i, however, a little different scenario is playing out. So, to surprising everybody, after Rod had been killing on Hawaii Island. All of a sudden, in April 2018, rod was discovered on Kaua'i in the northeast part of the island in the Moloa'a Forest, for those of you familiar with the area. And that was a surprise. Nobody expected rod to jump all the way across the archipelago from Hawaii Island here to Kaua'i, but it did. And so that was April 2018. As of yesterday, this is the presence of rapid ʻŌhiʻa death across Kaua'i. Now, it looks worse than it is, I will tell you, because not every single one of these dots represents a tree or even a number of trees. It just represents in red, the bright red, the one of the two species known as Ceratocystis leucoʻhiʻa. Its presence has been detected in these areas. The second species that we collectively capture or under the umbrella of rapid ʻŌhiʻa death is Ceratocystis hulioʻhiʻa. That's in the orange. And then there happen to be a handful of trees that are co-infected, meaning they have tested positive for Ceratocystis leucoʻhiʻa as well as Ceratocystis hulioʻhiʻa. Now, this total collection of dots here on Kaua'i represents about 447 trees here on Kaua'i that have been confirmed to have died of rapid ʻŌhiʻa death. Um, Here's how it looks annually. So 2018, you see we had 23 detections, 2019, 84, 2020, 108 positive detections, 2021, 84. And then you see the number drop off, and I have to make note of 2022 and 2023. There are likely trees that died during that time, but because of like post-COVID situations and staffing, funding, we were really not able to do as much work in the forest. So I would say we're back up to, in 2024 and 2025, probably what is a more accurate number and what's more accurately happening in our forests. That to say, we estimate there might be 2 to 3 times as many trees that have died here on Kaua'i of rapid ʻŌhiʻa death. And if we just take the high number, say 3 times 500, that's 1,500 trees, perhaps, that have died of rapid ʻŌhiʻa death here on Kaua'i. Thankfully, that is not a drastic number, considering ʻŌhiʻa is our most abundant native tree in the forest, and it is certainly not an exorbitant rise in the mortality of ʻŌhiʻa here on Kaua'i. So that's been kind of very interesting in the difference of how this disease is playing out on Kaua'i versus Hawaii Island. And during that time, since 2018 to now, we have done a lot of different work. It's kind of a three-pronged approach to responding to this disease. Those of you who recognize me or know me, you've probably seen me out in the community giving talks or doing programs or projects in a whole bunch of different ways. These virtual forest talk series, forest journaling, as well as school groups. We've done biosanitation giveaways. We've collaborated and continue to collaborate with organizations and businesses on the island. We've given away a whole lot of ʻŌhiʻa for people to grow in their yards. So there's a whole lot of outreach effort that's being done here, as well as my field crew at KISC and TNC and DOFA have responded kind of boots on the ground to this disease and managing rapid ʻŌhiʻa death in an effort to slow the spread. So we do drone surveys, we do quarterly helicopter surveys, we actually do in-field sampling of the trees, we do felling, we've done seed banking, and our latest, new, greatest project is the beetle repellent one, which you see in that photo on the bottom right of my colleague Lizzie. So that brings me to the third effort. I talked about outreach, management, and so there's a whole lot of science that's being done to understand this disease, as well as to figure out how the heck can we slow the spread and save our endemic ʻŌhiʻa. So these are just three of the areas that have the work that's being done. There's an ʻŌhiʻa disease resistant program going on. There's some surveys that are looking at the difference between areas of the forest on Hawaii Island that have a presence of hoofed animals. And by that, I mean pigs, goats, cattle, that kind of thing, versus areas where there is no presence of hoofed animals. And there is a drastic difference in the presence of rod. So where there are these hoofed animals, there's a whole lot of Rapidohia death, and then literally on the other side of the fence, very little Rapidohia death. And then the third area on this slide is the beetle repellent slide, and that is our guest speaker in that photo in the top left, Kylle Roy. And that's what we're here to talk about today. So I'm going to stop sharing so we can switch over. That should have stopped sharing. Great. To Kylle. And before I turn it over to Kylle, let me just give you a quick introduction. I've worked with Kylle now for about the past 7+ years. Really have enjoyed seeing the work that she puts into this project and this effort to save ʻŌhiʻa, support everything she does. But here's the official introduction. Kylle Roy is a forest entomologist for the Forest Health Protection Branch of the Forest Service, serving Hawaii and the U.S. affiliated Pacific Islands. She's based at the Institute of Pacific Islands Forestry in Hilo, Hawaii. She's a native Hawaiian, born and raised on Oahu, with a master's in tropical conservation biology and environmental science from University of Hawaii at Hilo, and a fresh PhD in forestry and natural resources from Purdue University in 2023. And with that, I'm going to invite Kylle to share. Welcome, Kylle. Thank you for saying yes.
Thank you. Thank you for having me. This has
My
been
pleasure.
amazing so far. I love that video.
Ah, good.
Um, yeah. Okay, let me just share. Get in there.
Getting there.
There we go.
I'll let you know when we've got the holes. There it is. Perfect. You're good to go.
Okay. Um, thank you everyone who came today, and thanks Kim for inviting me. Um, So the title of my talk is Back Off Beetles, How Repelling Ambrosia Beetles Helps Protect ʻŌhiʻa Forest. Kim came up with that. I can't take credit for the title of my talk. So this is actually kind of a new talk because it's specifically only talking about the use of repellents. Um, and I guess all of this work together was all part of this one special technology development program. Forest Service grant that we had. Okay, I'm gonna get started. So I don't have to talk about this too much because we had an awesome introduction, but so rapid ʻŌhiʻa death is caused by two different Ceratocystis fungi, Ceratocystis leucoʻehia and Ceratocystis hulioʻehia. Um, I don't think that I don't think the specific fungal names were mentioned, but leucoʻehia is the bad one. Hulioʻehia we think has been here for a longer time, so it's given that name huli just to turn over, and it acts more like a canker. Where leuku is ʻŌhiʻa destroyer, and so we're fortunate that leucoʻehia is only found on Hawaii Island and Kaua'i thus far. Whereas huli can be found on both of those islands and on Maui and Oahu as well. So uh, they are killing the most culturally and ecologically important tree, ʻŌhiʻa lehua. That video did a really awesome job talking about how important ʻŌhiʻa is. So it holds up this slide a little longer. I always feel bad when I only have enough time for this much of a slide, so that was great. So this is the disease cycle that Mark Hughes had illustrated, and so this is as of 2025. Basically, the way for rod to spread, ambrosia beetles are one of the main vectors, and so they dig into trees, push out frass, and in that frass there can be a viable inoculum that can move in the soil, in the wind, on the hooves of feral ungulates. So if you notice in this disease cycle, we have the pigs off to the side. So they will be added to the disease cycle. We're just waiting on publication. So we know that that pigs can carry viable Ceratocystis propagules, and there's ongoing work by Sarah Knox and Kylle Roy that are looking into how often they are rubbing up on trees, and if they can directly vector ceratocystis. Um, but I feel like there's no doubt that they can definitely carry frass on them or sticky spores too and take them to a healthy tree and infect it that way. So there's these two things going on, the beetles and the feral ungulates. The beetles can also directly vector ceratocystis. So they're attracted to stressed trees. They may look healthy to us on the outside, but they could be stressed from nutrient limitation, drought, fire, wounding. Um, and then they can go from a tree that has rod and then infect a stressed ʻŌhiʻa tree. So all of our work has found that there are 4 invasive ambrosia beetles that can vector Ceratocystis leucohea and Ceratocystis filiohea. Um, there's Xyloborhinus saxonii, Xyloborus affinis, Xyloborus bispinatus. You can see there's a red X over ferruginous. We're in the process of changing it, so in the history of entomology in Hawaii, it's actually been called by the wrong name. And so Ellen Dunkel has been leading a study where we looked at historical collections and new collections and DNA, and found that actually we have Bispinatus and not ferruginous. And knowing that was important because that beetle is the one that actually produces the most frass, and so it's kicking out the frass into the environment, and they are limited to the lower portion of ʻŌhiʻa trees. And then the last beetle is Xyloborus perforans. So what unifies all these beetles is they're all in the Xyloborini tribe. They have this haplodiploid biology where a single founding mother can start an entire generation, start and there, so it makes them really effective invasive species. So I went over this a little bit, the 2 ways that the beetles can be involved with the spread of rod. So these are 2 different studies that we really went through Koch's postulates and then also Leach's rules of insect transmission to be able to show that frass and ambrosia beetles directly are a big part in the spread of rod. The key takeaways were spores in frass. The spore type is called a chlamydospore, so that's just a resting spore. And we found that it could survive for at least 6 months in cool, humid environments. And that was just the limit of the study, so it could likely survive much longer than that. Um, and then for the direct transmission, we found that some species, such as Xyloborus affinis, are more likely to carry viable propagules on their bodies. So, in order to control these ambrosia beetles, you can use chemical ecology. And so you can manipulate them using attractants or repellents. We refer to them as semiochemicals. We've tested different attractants. Some examples are ethanol and ethanol methanol, different mixtures of that combination. And what that is doing is really simulating that the tree is a good place for the beetles to plant their ambrosia fungi. So simulating that stressed tree that they have adapted to come in and attack. And then different repellents that we've tested. They're from this company called Iskatech. It's called both the repellents that we've tested are in this splat formulation. People always ask me what splat means. And it really and the word that comes after splat is very important because that's usually what is in the formulation. So splat is a specialized pheromone lure application technology. And so you can see my pointer. It's this waxy matrix that just binds onto the tree and you push it out of a caulking gun. And then yeah, it just it adheres to the tree and then it biodegrades within 3 to 4 months. So it's natural. So we tested verbenone, and that product is called Splat Verb. And then we've also tested methyl salicylate, and we tested a product called Splat Beetle Guard, and that has 10% verbenone and 10% methyl salicylate. And in this particular system, these two scents are signaling that the tree is decomposing and it's not a good place for these beetles to plant their ambrosia fungi, and they don't want to. It's basically telling the beetles, don't come here. It's not a good place for you to survive. Basically, it's past the prime stressed period where that these beetles like to inhabit. Um, okay. So the first step, I'm kind of just going to walk through all the steps that we got through to get to where we are now, applying Splat Verb across the state. Um, So the first step was what we call a trap shutout study. And so that's testing lures. So pictured here, it's a lure, ethanol lure, and then comparing that to the repellent. And so we did this with Splat Beetle Guard, which is the verbenone and methyl salicylate, and then Splat Verb, which is just verbenone. Um, and we found that, first of all, ethanol was the best attractant for all the rot-associated beetles. We also had other studies where we found when we wounded and stressed ʻŌhiʻa, they emitted that ethanol scent, really showing and that also increased beetle attack, so really showing that relationship with the ethanol. Um, and then we also were able to show that, both products, Splat Verb and Splat Beetle Guard, were effective repellents in the trap shutout studies. And that's published in this paper here. So Beetle Guard has a temporary experimental registration with the EPA. So when we moved on to actually testing this directly onto live trees, we only tested Splat Verb. So this study published in Trees, Forests, and People in 2024. So we wanted to test Splat Verb directly on healthy ʻŌhiʻa trees and also on trees that had rod. So the idea is to be able to protect a healthy ʻŌhiʻa tree from getting rod, and then for trees that already have rod to prevent further beetle attack and frass production, and then therefore the spread of rod. We also tested two different rates. We tested the low label rate, which was 72 grams, and then the high label rate at 108 grams. And in the lab, we also wanted to know how long this active ingredient was, we could detect it. So we quantified the abundance over time using gas chromatography, mass spectrometry. That's what these are pictured here. Oh wait, let me show this to you too. Because people always want to know. So this is what we did in the study. We had sticky cards like right underneath where the repellent was. So getting an idea of beetle attacks onto the sticky cards, and then it's really easy to apply the Splat Verb just with the regular caulking gun. And then you can just see the Splat Verb dollops there. Okay, so now we're looking at the cumulative numbers of, well, the average, I guess, number of beetle attacks or landings on trees that were healthy ʻŌhiʻa, that were treated with the low dose of Splat Verb and the high dose of Splat Verb. And so all the graphics for this study, all the controls are always going to be in brown, and then the light green is the low verb dosage, and the darker green is the high verb dosage. And you can see circled in red, both dosages were effective at reducing all of the rot-associated ambrosia beetle attacks. And now this is looking at so there was two trials. So now we're looking at this at the beetle attacks over time because we wanted to know, how long the splat verb is effective for. Um, and we can see in both in 2022 and 2023, the high dose was effective through week 12, and then the low dose through week 14. So that's pretty good. And then I'm not showing the lab results of the splat verb by itself when we're looking at it over time, but we did see that there was a huge drop off in the active ingredient within the first 8 weeks. So it was impressive to see it lasting this long. Um, despite the active ingredient really dropping off. And then now I'm going to go through the data for the rod affected trees. So we're showing this with this brown ʻŌhiʻa tree. Um, and so it was a little bit different for the rod affected trees. In 2022, only the high dosage was effective. Well, I mean, it still reduced the number of beetle attacks, but the high dosage seemed better, and it was significantly reduced the number of beetle attacks. But then in 2023, both low dose and high dose significantly reduced beetle attacks. Then when we look at those data over time, um, we see in 2022 it was a little wonky. The low dose was never significantly different than the control at reducing the rod-associated ambrosia beetle attacks at any time point. But then when we see in 2023, it was actually effective through week 12. And so what we think was happening here is that the rod trees are producing a ton of volatiles, and so we think that we needed the higher dosage to kind of block out the rest of that smell that's coming out of the tree that's attracting the beetles. So let me see how I'm doing on time. Okay, doing good. So putting all of that together, um, so the takeaways are the low dose, which is 72 grams, which costs about $13 a tree, is the most efficient and economical treatment in healthy ʻŌhiʻa. And then in trees that have rod, the higher dose of 108 grams may be the better choice. And that does cost more. It's about $20 a tree. And especially on Kaua'i, putting these two things together, where there's new rod outbreaks, you could treat the trees that have rod in the center and then treat a radius of trees, of healthy trees around it. And so what Kaua'i is doing is treating the 10 meters of healthy trees around the rod trees. And based on our looking at the data over time, we saw that healthy trees, reapplication might not be necessary since it's effective for 14 weeks, but for trees that have rod, the data were not so clear. So between like 10 and 14 weeks, reapplication may be needed. And we're saying that so this is based on there's kind of a peak season where there are tons of ambrosia beetles. So that's between May and September. And so if you had to only treat one time, that's when you would do it. But I'll get more into what's actually going to happen on the ground. And then moving into this other study that was just published, Dio Mikros did this for his master's from University of Hawaii at Hilo. He's back working with me again. And so it's the first time I'm presenting his work. And so it was just published in Environmental Entomology. That's him. Of course, there's a picture where he's covering his face. But you'll get to see a picture of him at the end of this presentation. So he tested splat verb and splat beetle guard. So that verbenone plus the verbenone and methyl salicylate, those two different products, as a post-felling management strategy. So before we had the use of repellents, the only management we really had was fencing, which is great. And then also felling trees that have rod, and then tarping them. And tarping can introduce, you're introducing trash into the environment. You're also creating mosquito habitat, which is not great for the native birds and avian malaria. And so we wanted to know if we could use repellents to treat trees after they've been felled. And so he looked at it in 3 different ways, seeing if the repellents will reduce beetle attack, reduce beetle emergence, and also frass production. And I guess I'm going to point out here, so this is like a felled tree, and this particular one wasn't treated with repellents, but the repellents we would place in each corner, and then these are actually marks of all the individual beetle galleries over time. So there's tons of beetle attack. So looking at the cumulative beetle attack, so both the repellents in this series of figures are shown in 2 different green colors. The splat verb is that darker green, and the splat beetle guard is the lighter green, and then all the controls are in blue. And so then there was 2 different trials that were done. And so we can see here that both repellents significantly reduced beetle attacks. We do see that sometimes beetle guard was more effective than splat verb in trial 1. Then when looking at the beetle attack over time, this is important because we wanted to know if the trees that once they were felled and treated, if they needed to be retreated. And you can really see at the end, all the beetle attacks subsided by week 10. And so this was what we really used as evidence, is you don't need to go back and retreat the trees, because once they're felled, they're drying out and it's making it less desirable for the beetles, so they're not going to be attacking them anymore. And so now we're looking at the beetle emergence data. This was weird and kind of all over the place. So we can see that beetle guard was effective at reducing beetle emergence in the first trial. It was kind of all over the place in the second trial, and we think this might just be a factor of we're like, okay, it works sometimes. But it's kind of more of a factor of we're looking at the beetles that were already in the tree, so it really depends on the specific tree. And so if you have a single bolt, you can get tons of beetles from a single gallery. You can get like 50 plus beetles from a single gallery. And so it's really hard to interpret these data. And then looking at the frass production, you can see both repellents significantly reduced frass production in the first trial. And then again in the second trial, is like not so clear, but there was less frass production in the bolts that were treated with verbenone. And again, this we think is attributed to the different timing or the seasons when we actually felled the trees. But together we can definitely say it does reduce frass production, and that is really what we're trying to do when you're felling the trees, is you want the trees to not be, you don't want the beetles to be coming to the trees anymore and producing frass and reproducing. And so there's more beetles that can vector ceratocystis in the environment. So reducing all of that. So altogether, putting that all together, the takeaway is a single application of splat verb or splat beetle guard can reduce beetle attack, beetle emergence, and frass production. So now that we did both of those things, we put them together and updated this rod management flowchart. And so this was made way back in 20, I want to say like 2016, 2017, by David Benitez, who at the time was the Hawaii Volcanoes National Park ecologist. He's moved into a higher position now. Um, and so we updated this to include treating with repellents. And so you can fell a tree, and then if tarping's not feasible, treat with a repellent, or if you want to leave it standing, also treat with a repellent. And then we included information with the EPA label number. And I believe the rod management flow chart is now, so it was in that publication, that Dio, this publication. So it's in here. And I think that it's also on the Rod website. And JB's here and he can say in the chat. I'm pretty sure it's up there now. And so the next step, as of October 2025, Splat Verb has a special local needs label. So you can purchase it and use it in Hawaii. And we did something different. So people have been testing Splat Verb for many years, over 10 years, for other uses. So it was originally developed for mountain pine beetle in the continental US. And people have been testing it. So Dr. Mark Wright has been testing it, and Angel Aspis Doria has also been testing it. And I was like, hey guys, let's get it. I'm gonna get it registered for ʻŌhiʻa. Let's also get it registered for MacNut and coffee. And so yeah, now people can use it in MacNut and coffee as well as in ʻŌhiʻa forests. And that's good for the next 5 years. Um, and so how's the timing? Timing's good. Um, so then what came out of this is Kelly Harshman. I'm sorry, this picture is so pixelated. I took it off the internet. Um, this is Kelly. She works for DOFA in Kaua'i, and she put together this splat verb protocol. And I came out to Kaua'i in December. And then this is the crew that went out. Rob Hoff was also there. Unfortunately, Callie couldn't make it, but we were able to, we were able to apply splat verb and go through the protocol with everybody there. And at Waipo'o Falls, if people recognize that. And then we made edits. I sent the protocol around because we want it to apply to the whole state. So I sent it to various managers across the state, and people gave input, and we put it together. And now it's going into each individual island's strategic response plan. And with that, I can take any questions. And so here's Dio in the Vans t-shirt, and then Ellen Dunkel has also been a huge contributor to all of this work. And these are our emails if you have any questions that you're too shy to ask right now.
Thank you, Kyle. And let's all show our appreciation, using our fun emojis at the bottom of your screen under the react button. There they come. There's the applause happening. Oh, yeah. Coming in fast and furious now. Thank you, Kyle, for not only this presentation for today, but the year's worth of work that I know went into this. I'm going to remove our spotlights, and if you would maybe stop sharing so that we can all join back together in the homeroom, so to speak. And oh, look, the applause just keep coming. I'm loving it. Yeah, thank you. It's super exciting. We're excited to be able to utilize this tool to help save ʻŌhiʻa. mean, is what it all comes down to. So, again, if you have questions, drop them in the Q&A. If you can't find the Q&A because you're on your phone or something, put them in the chat because it's not too crazy in there today. But we do have a question for you, Kyle, from Andrew in New Zealand. He asked, and he dropped this in early in your presentation, so you may have answered his question as you went on, but you might want to pull up the Q&A because I don't know that I'll be able to pronounce this word correctly. But have you tried quercevorol as an attractant?
Um, that is an excellent question because well, my answer is no, we have not. But so that's the attractant for Xyloborus glabradus, pretty sure. And actually, Mark Hughes and I have been talking about getting some and seeing, putting it out, especially on Kaua'i because there might be Stratocystis leucohea infecting camphor trees, and so we kind of just wanted to go out there and get an idea, figure out if they are, yeah.
Yeah.
Anyways, figure out if glabradus is here, because it hasn't been done, and we are thinking about that. Interesting question.
And Andrew, if
Interesting and very specific question.
isn't it? I
And
suspect Andrew's.
I hope I'm correct too, and I just didn't go on a tangent and tell you about something
And Andrew, feel free to go unmute
else.
yourself if you want to follow up with that. Oh, there he is.
I was just gonna, yeah, I guess all people claim it's attracting to all kinds of different ambrosia beetles, so I was just kind of curious. We're gonna put out just a handful this summer in a couple of weeks and see what happens here in
Okay.
New Zealand. So I mean I can let you know, see what we find.
Yeah. Let me know.
There's definitely plenty of Saxonia coming to our ethanol lures anyway.
Yeah, I would love to come to New Zealand and see what you guys are doing.
Hey, Kyle, why don't you drop your email address, your preferred email
Okay.
address in the chat? That way Andrew will have it and you guys can follow up. And I would love to see that happen. That's fabulous. Um, let's see. There is another question in the chat from Bill P. Is anyone looking at what's happening to native species that feed on sick, dying, and rotting ohia?
So the answer is well, yes. So there's one native ambrosia beetle that feeds on sick, dying ʻŌhiʻa, and so they're Xyloborus similis, and that's actually what is in our logo. But they're endemic to Hawaii Island, and we do have evidence that they're disappearing because they occupy the same niche as Xyloborus bispinatus. Um, yeah, they're I would say they're probably the only insect that is preferring the dead ʻŌhiʻa. I'm sure, I mean, there's over 400 endemic arthropods that love ʻŌhiʻa. And yeah, and but the one that is for sure disappearing is Xyloborus timilis.
Hmm. That is related to a question that I had somewhat. Speaking of the different species of ambrosia beetles, I think you mentioned that uh, a finnis is more likely to carry the viable um, spores on their body. Is that correct? Did I get that correct? And my question is, why are they more likely to carry the viable spore than another ambrosia species?
So we think it has to do with their behavior. Maybe they're not cleaning themselves as much. And they are also hairier than the other
Mhm.
beetle species, so we think the sticky spores are more
Ah,
likely to get stuck on them. Yeah.
See, I knew there would
Excellent,
be a
excellent
perfect
question.
I knew there would be a perfectly understandable, explainable answer. So thank you for that. I'm going to jump back to the chat because Brad has a question. Do you use drones with IR sensors to find infected trees? And I realize that is not your exact area of specialty, but I also know that you, like several other people in the room today, are on the ROD working group. And so you might be able to answer that.
Yeah, so I don't know what IR sensors are. Um,
Infrared is what I would interpret that.
okay, okay. Um, so Ryan Perroy has looked into infrared um sensors for it was like a little project that he did just in the lab. And they weren't really seeing any differences, but it seems like since he's done that, technology has changed
Mm.
and gotten better. And we are part of this bigger NOAA project where we're hoping to actually use drones for IR, and also looking at the volatiles of the entire forest and
Oh.
hopefully being able to detect individual trees too that have rod and don't have rod.
Oh, so am I understand that you're saying we're going to be using potentially drones to collect volatiles?
Yeah.
You're amazing. See what I mean about the cool stuff you're doing? And
I
so,
would say those, Ryan.
well, but
I'm just
you
along
guys are,
for the ride.
that's going to be a partnership, though, I would
Yeah,
imagine,
yeah,
because
yeah,
you're
it
doing the
is.
volatiles, collecting the scent profiles and what have you. Um,
Brad did a follow-up. Brad, I don't know much about drones. I'm sorry, that was my extent of my answer.
if there's anybody else in the room that's in our working group that can maybe speak to that in a little more detail, Brad's point in question in the chat, feel free to jump in and unmute yourself. And if not, at any point, if not, I do have a follow-up question myself. Um, I'm gonna guess that the research that you did with the beetle repellents was on Lukuohea trees. So, just for everybody else, background, there are actually two species that fall under the Rapidohea death disease umbrella, if you will. Two species of fungal pathogens. One that we refer to as Ceratocystis leukohea, Kyle said is the destroyer of ohia. And then the other is Ceratocystis huliohea, which turns over ohia. It's not quite as deadly. Uh, so my question, Kyle, I'm assuming that your studies were on Ceratocystis leukohea, and if you think there might be any difference in how the beetles interact or would respond to this beetle repellent, whether it was a tree that died of Ceratocystis leukohea versus Ceratocystis huliohea.
Yeah, so that's a really excellent question. We actually tested it on trees that had leukohea and huliohea, or trees that had both.
Oh, good.
Um, we didn't have enough sample size to look if there was differences in beetle colonization on leukohea versus huliohea. I tried. Um, But so the reason why we used both was because when we were doing these studies, we had experimental use permits, so we were limited to, gosh, 10 acres, I think. So first, like the second study when we tested beetle guard, that was limited to a quarter acre since we're using something that wasn't registered with the EPA, and then we're able to get 10 acres. And so it's hard to find enough rod trees in the perfect condition anyways, with like 10 meters apart from or 20 meters apart from other trees or whatever in all of our plots. So we did use both leukohea and huliohea. Um, and unfortunately you're unable to actually answer if there was a difference. But to answer the rest of your question, we've looked at how the beetles respond to the volatiles of leukohea and huliohea separately. And so we had different results. We saw that Saxonii, the one, there's tons of them. They're kind of everywhere. Um, they were actually repelled by ʻŌhiʻa. So we're starting to think that that's a line of evidence that, there's just a ton of them.
Hmm.
They're not, maybe they're not a big deal with broad spread. But the thing about them is that they will colonize the whole bowl of the tree. So if they're producing frass that has inoculum, that will get blown in the wind. And
Mhm.
they're smaller beetles, so it's smaller particles too that can move. And then we saw that Affinis and Bispinatus were both attracted to leucohea. Um, and we also, on top of that, wait, let me finish this answer first. And then we saw that um, Perforans and Bispinatus, again, were attracted to Juliohea, and that
Mhm.
it might lead to also where they're from, and also where Leucohea and Juliohea might originally be from as well.
Well,
Um, and we do see, I mean, if you look at the numbers at the numbers of like co-infected trees, I have a hypothesis that it has a lot more to do, co-infections have to do with ambrosia beetles, because when we're doing studies, we see co-infections all the time because we're doing ambrosia beetle studies. Um, and on top of that, so we're also looking into if the beetles can survive on the ceratocystis fungi. So they're fungus farmers. They have like yes, they have a symbiosis with a fungus that they farm. But they're not farming, or what we think is not the main thing that they're farming is not ceratocystis. But how are they surviving in trees that have ceratocystis? So you would think that their ambrosia fungi is um, being potentially being outcompeted by the ceratocystis. But we do see certain species that tend to be in the trees much longer, like Bispinatus. And so um, new work that Ellen Dunkel has been leading is looking at if they can in the lab survive on Ceratocystis leucohea or Juliohea. And right now, yeah, it's pointing towards, yeah, it looks right now she's only looked at two species, the Finnis and Bispinatus, and it's looking like Bispinatus survives pretty dang well on Leucohea. What
okay.
does that mean? They're more likely to be vectors.
So in my takeaway, it's complicated.
Yeah, it is
So
complicated.
there's a lot, like looking at the life history and the biology of the different species of the beetle, and then the different fungal pathogen and where its origin might be. It sounds like there's a whole lot of opportunity for more science for you to run.
Yeah.
Gonna keep you busy. Yeah.
Yeah.
Let's see.
Are
Uh,
you wrapping up?
Go ahead,
Or
Brad.
can I ask a question?
Yes.
Here's the deal with drones. I, and a little short background. For 17 years, I served as a primary contractor to the Forest Service, giving them GIS data and
Okay.
data sets and applications. So, um, my question about drones is if you put an IR band on a drone with red, green, blue that's normally on a drone, then you should be able to recreate your forest in 3D from sequential drone photos and make a 3D representation of the forest. And be by through those four bands, you can tell trees that are not healthy and go in and look at them and determine whether you need to apply a treatment to a specific tree in the forest. So
Okay.
I'd love to work with you all because I've got 40 years in GIS and I'm on the island on Kauai.
Oh, you are.
Oh,
Great.
amazing.
Uh, so Brad, I want to point you to the chat because JB Friday, who's been involved in this response to Rod since it was first started to being discovered here or over on Hawaii Island, he's responded. He's made a comment in the chat. We
Okay,
have,
and I can't figure out how to get back to the chat.
oh, okay, JB, unmute yourself.
Hey, Brett, we have been using a lot, and I'm not the remote sensing person, Ryan Peroy at ʻŌhiʻa has done most of that. Your earlier question, currently our monitoring is just using visual wavelengths, but there was some research done on near infrared, and they were able to determine trees that were going to get that already had the disease but were pre-symptomatic. So they are able to determine trees that 2 weeks later developed visual symptoms from using a near infrared. In general, the monitoring has been done just by the visual because, again, when they do first turn, the whole tree turns is quite suddenly, quite easy to pick up in it's easy to pick up just in the visual spectrums. What has been a bottleneck is these are visually analyzed. So a student or a technician sits there looking at all the images to pick it out. There was one master's student who tried to look at machine learning. Can we automate it? And the basic answer to that was, well, not quite yet. It's not quite there. I mean, someday the technology will be there to run it through the machine learning so it can automatically detect those.
Yeah.
And the other thing that people are working on is moving from drones to helicopter imagery to satellite imagery, and being able to cover more area. And then, of course, if you can do it with satellite imagery, you don't have to do all the expense of going out and flying.
I got it. And with drones, though, you can get really high resolution. If you suspect someplace, you can go out there and create a 3D landscape using points in the RGB and create a 3D landscape over what you've flown. And you can find specific trees very easily. And I worked for NV5. It was photoscience at one point in time, but they now have technology to do the AI assisted stuff. You can find the height and the biomass of every tree in the forest just using drones.
Okay, this is a great conversation. JB, do you think maybe we should connect Brad with Ryan Peroy?
Sure.
Okay. Brad, I should have your email if it's the one that you used to sign up for today's Zoom, and I'll reach out to you and make that introduction. And
Fabulous.
I do have one more quick question, JB. Stay on the line if you will, because I think you might be able to answer it. Does anyone out there know if there is DNA evidence of past epidemics in ʻŌhiʻa or any other native species?
There was a past epidemic in the 80s in
Mhm.
ʻŌhiʻa. I don't know what it means by DNA evidence. I mean, there was lots of work that was looking into if that was caused by Phytophthora, and ultimately the scientists came to the conclusion that it was cohort senescence, so trees growing up in a cohort together and then running out of nutrients and dying and then succumbing to other things. But thanks to
So,
that,
Kyle,
yes.
and that is what the ecologists came to, because basically everybody looking at ecologists, it won't surprise you to know that Tom Harrington thinks that's completely wrong, and they just didn't pick up the pathogen that was there. So I don't think that is settled. I think the ecologists are satisfied with that, but I find it hard to believe there wasn't something else going on. But nothing else there was no smoking gun. Of all the pathogens they found, nothing was like unusual, unlike the Ceratocystis, where once we found this starting to die, there was a new pathogen in the block.
Yeah, well, from what I read, it seems like it was all hands on deck. Like right, all the pathologists, all the entomologists tried to test everything, and they couldn't find anything really that they really thought couldn't come up with a conclusion. And so cohort senescence, I believe in it.
Well,
But to answer the question, though, that yes, there have been large episodic dieback issues. This is the first time that we've seen one with a non-native pathogen. And, for us, this is echoes of Dutch elm disease and chestnut blight and other such forest pathogens.
great. So thanks, JB. Thanks, Kyle. Thanks, everybody in the room who's still here with us for showing up and for caring, right? So I invite you to continue doing that. And I will be following up today's session with a video recording. I'll send the link in the next day or two once I get all of that edited and uploaded. And don't forget, our next Forest Talk session is going to be with my colleague Halen Chalk, and her title is From the Mountains to the Sea, The Importance of Limu in Connection with Watershed Health. And that is going to be on Wednesday, March 25th at 10 a.m. And you'll be getting notice of that with the registration link to come. Meanwhile, once again, thank you, Kyle. Really appreciate this presentation as well as all the great work that you're doing. And to everybody else, ahui ho. See you next time.